Briefly today, we want to discuss some of the mechanisms that underlie embryonic development. How does a cell know what it is and what it actually must become? We've discussed the idea that cells are undifferentiated in early development, meaning they don't know what they're going to become until they get the chemical signals to do it. There are a number of chemical signals that will come into play. We'll talk about morphogens in another video, but what I want to think about today is actually location, location, location. Ultimately, when cells start to differentiate based on directions of chemical signals, they will actually induce cells around them to become similar cells or different cells based on their cues and different chemical cues as far as hormones go. Now, when we look at embryonic cell development, we can start to think about this limb bud idea before that we've talked about in other videos. We'll talk about it with morphogens, and a morphogen is just a change. So this is a chemical that causes a change. And in particular with limb buds, we'll talk about something known as sonic hedgehog in the morphogen lecture. But what I want to focus on is how this actually induces different development. So when we have this ZPA, the zone of polarizing activity, what does that actually mean? That means that there are chemicals produced in this limb bud, what will become the wing of the chicken. There are chemicals there that say, okay, we're going to be a limb. In particular, we're going to be a chicken wing. If I want to be a chicken wing, how can I get this to develop properly? And what we're actually focusing on is the development of this very end of the chicken wing, known as the wing tip. For those of you that are buffalo wing connoisseurs, this is actually a part of the wing you never see. This is the drumstick part of the wing, and this is the flat part of the wing in culinary terms. But when we look at wingtip development, normal chickens have one wingtip per wing. But if we transplant some of those chemicals from here to here, we can actually get two wingtips to develop. Why is this interesting? Well, we've isolated a chemical that causes one particular phase of development. That's critical to understanding what could go wrong in development. So keep in mind, when we talk developmental biology, we're not just interested in what goes right. We're interested in showing how things go right so that we can then determine when things go wrong. So what we're looking for are these genes and proteins to actually interact. So we're going to have this signal come from the DNA, go to the mRNA, as we've talked about before, and then eventually to a protein. So what we're interested in is who controls most of this. At what point does a cell say, yep, this is what I need to become? In particular, when we're developing more complex things like organs and tissues. So we have to start thinking about actually sculpting the body, as I have here. What's actually going on to create not only a three-dimensional body plan, but then to take that body plan and make it look human. So characteristics that we're accustomed to in humans, 2 arms, 2 legs, and so on. Those primary characteristics, how do we get what started out as a ball of cells, remember? We were just all a big ball of cells. How do you take that and make it look like you? We're dependent on a couple of properties, cell adhesion being the first. What does adhesion mean? Yep, to stick together. So cells want to stay in contact with one another. I'm less concerned about all of this. Okay, what I want you to focus on is that cells want to touch other cells. This should make sense to you. You have very few large gaps in your body. Now, we can talk about your mouth and things like that, but you have very few gaps. Cells want to be into contact with one another. We also have to deal with something known as cell death, or apoptosis. This is known as programmed cell death. This is going to be critical in development, because what we look for is taking that ball of cells that we have, right? And, all right, if we're going to get that to turn into a person with arms and legs that are separated, how are we going to get that separation? Why don't you just look like an elongated ball? Okay, so what we have to think about is this apoptosis, or programmed cell death, coming in and actually eating away at the cells in the middle, killing them, so that you can end up with two legs. And then we'll come in with more cells and eat out those other cells, kill those other cells, so you can end up with an arm, and we'll have some here that are going to form a neck for you. This might not look a lot like you. But we're going to get to the point where that's actually what you do look like in a general sense to start out. What is apoptosis? This is simply programmed cell death. It means that you have a digestive cell. This is actually going to be part of your immune system that is going to go in and target any cell that has been flagged for death. So your developmental hormones and chemicals and morphogens are going to say this cell must die. And when it does, this cell goes, dinner, and will engulf or surround and eat that dead cell. So this cell is now this cell. Okay? And you see eventually we just totally engulf and digest that cell so that it's totally gone. Where do you think we see this in you? Well, I gave you hints that it makes your arms and legs and neck. But one prime example are your fingers. These are your fingers early in development. You kind of have a flipper, right? The swimmers in the audience might wish they still had those. But if you actually want defined fingers, these spaces in between are formed by apoptosis. So, before we talk about chemical signaling and morphogens, we need to keep in mind that cell adhesion and apoptosis will be critical functions in development to make sure that not only are our cells in the correct place, but that we'll have good covering and a solid body form when all is done.